Plants feel the sun, taste the sun.
In this video, we are going to discuss photosynthesis described by Hunter, Shayna, Kristen, Jackie, and Nick.
So what is photosynthesis? Photosynthesis is a process by which carbohydrates are built from sunlight and carbon dioxide taken from the air.
These carbohydrates are then used as a structural component of the plant, as well as a source of energy used to
We chose the creative approach of candy because candy is fun and easy to manipulate the structures of photosynthesis.
This approach helped the group better understand photosynthesis because we got to see our 3D candy structures act out the movements of photosynthesis.
Plants use their leaves to make food by taking in the energy from sunlight. Plants also give off oxygen that exits their leaves, and this keeps us alive.
Now we're going to take a closer look at a plant. Here we have multiple plant cells. Inside the single plant cell, there are many chloroplasts. Now let's take a closer look at the chloroplast. Inside the chloroplast, there are many different thylakoids. To see how photosynthesis works, we are going to zoom into the thylakoid membrane.
A water molecule comes in and binds to the enzyme photosystem 2 at the reaction center. The sunlight comes in and splits the water molecules and uses the electrons from the water to replace the electrons removed from when the sunlight photons hit the reaction center. The photon captured by the chlorophyll within the photosystem 2 bounces between chlorophylls before it finally reaches the reaction center where it excites the electron.
The electron is now excited, and the now excited electrons move through the enzymes on the electron transport chain. The energy from the electron moving is allowing the pumping of hydrogen in the lumen.
Now we're at photosystem 1, and the electron from photosystem 2 is going to get excited even more.
The newly excited electron then proceeds through complex FD as it makes its way into NADP reductase. The electron in NADP reductase is passed to an NADP, which creates an NADPH electron intermediate carrier.
As this process continues to occur, a proton gradient gets established in the lumen.
Gradient has built up hydrogens begin to flow through ATP synthase, where ADP is turned into ATP.
Next, we enter the Calvin cycle, which takes place in the stroma.
The Calvin cycle starts with RuBP. Then carboxylation occurs, in which carbon dioxide is added to the RuBP. After carboxylation, reduction occurs, and reduction ATP and NADPH from the electron transport chain are used to make G3P. Once the G3P is made, the leftover carbons go through regeneration. In regeneration, ATP is used to regenerate RuBP. The G3P created is later turned into sugar.